Okay, so hello everybody. My name is Marysia. I am a sophomore in high school. I'm from Poland, and I'm very interested in biology, specifically genetics. And this is why today we will be talking about an insight into CRISPR-based therapeutic targeting of ADHD. So first of all, I would like to start off by asking you and yourself a question, which is, what do you associate ADHD with? When you think about ADHD, there are many things that can come to your mind. For instance, being messy, being lazy, having very hyperactive activity and being very hyper, and maybe even being impulsive. That is something that people with ADHD deal with. What ADHD is, it is a neurological disorder that is characterized by inattention and impulsive behavior. This also correlates to having problems concentrating or forgetting things in general, something that people with ADHD struggle with. And in order to treat those things, we use ADHD medication. The problem is that many people find ADHD medication very ineffective. For instance, when it comes to children, 80% of children find ADHD effective, whilst 20% don't. And this, for instance, includes my cousin. My cousin struggles with ADHD and she finds ADHD medication very ineffective, which is also a factor which motivated me to find new solutions in the aims of treating ADHD. Due to the fact that ADHD has a 77 to 88% irritability rate, it means that genetics plays a very vital and crucial point of whether you have ADHD or not. It basically means if you have a very high irritability rate of a neuro-associated disorder, it means that genetics play a very vital part of whether you have it or not, and we can identify which genes and which part of the body is responsible for the disorder. Then, what this means is that we can use genetic modification or genetic-based approaches and therapies, such as CRISPR-Cas9, in order to treat these neuro-associated disorders. And this, of course, led me to writing this research review article and becoming a Polygen Scholar of 2022. So what was my research review article actually about? What it encompassed was the solution of using CRISPR-Cas9 in the aims of treating ADHD. What CRISPR-Cas9 is you can describe it as a pair of molecular scissors. Basically, it's a tool that we can use and researchers and scientists have been using for quite a bit now to manipulate genes and manipulate the genetic material of our organisms, not only humans, but also other species. The obstacle that we stumble across if we would like to use CRISPR in order to treat neuro-associated disorders such as ADHD is the blood-brain barrier. The blood-brain barrier function is to isolate and protect neural tissues, controlling the entry of molecules from blood to the brain. As a result, the blood-brain barrier hinders attempts to deliver molecule-based therapies. What this means is that if we want to deliver a CRISPR-associated reagent, if we want to deliver the system to the brain in order to treat ADHD, we need to encapsulate it in some sort of capsule, or we need to implement nanotechnology in order to get it. Otherwise, it's impossible because our BBB is like, wait a second, this is not a molecule that usually passes through here. Like we cannot let it pass through, right? That is why it is a very big obstacle that we need to overcome. And it's an obstacle that we need to overcome because CRISPR continues to revolutionize medicine, and it has a very growing potential in order to treat a range of genetic diseases such as neurodegenerative diseases and blood disorders. So achieving effective delivery of CRISPR reagents in the brain would not only benefit therapeutic targeting of ADHD, but as I said, many other neurologic and psychiatric disorders. So what we would have to target in order to treat ADHD would be the D4 dopamine receptor. The D4 dopamine receptor is a synaptic neurotransmitter receptor that's responsible for an area of the brain that regulates emotion and complex behavior. When a person has ADHD, this area of the brain is disrupted. ADHD medication targets this area of the brain, and we will do that with CRISPR as well. But the difference here is that CRISPR is very precise. And by it being very precise, it means that we could get a lot more precise and more efficient results and efficient ways of treating ADHD. So there is more information to this. So during the production of dopamine, which is the neurotransmitter that that when a person has ADHD, they have low levels of this neurotransmitter. And during the production of dopamine, there are non-receptor genes involved, referred to as COMT. This is basically an enzyme that produces dopamine and transporter SLC6A3. And basically, we can use CRISPR to target these genes in addition to the dopamine receptors. This is the COMT here you see on the image, and here is a neuron. What you can see here are the red circles, and these are referred to as dopamine-containing vesicles. What these vesicles have are these dopamine neurotransmitters. As I said, when a person has ADHD, they have low levels of these dopamine neurotransmitters. With CRISPR, we will target these vesicles in order to increase the levels of neurotransmission and the concentration of these neurotransmitters, and therefore this would decrease the risk of ADHD, right? So with CRISPR, we would just have to target these vesicles, and then more dopamine would be released into the synaptic cleft, and that would, of course, bind to the surface of the postsynaptic neuron. So in conclusion, targeting the dopamine D4 receptor and dopamine associated genes could not only cure ADHD, but also help with studying and having broad implications in other neuropsychiatric disorders such as Alzheimer's disease and depression. And one of the biggest drawbacks now of CRISPR-Cas9 is in the treatment of neurologic illnesses is delivering the CRISPR-associated reagents through the blood-brain barrier. As we said, the blood-brain barrier basically blocks anything that it doesn't associate as a molecule that usually passes through. And this is why we would have to encompass this CRISPR-Cas9 system into a nanocapsule or anything that now we have advancements in nanotechnology, and anything that would imitate a molecule that would usually pass through the blood-brain barrier. And now there are promising strategies to overcome this limitation of overcoming the blood-brain barrier, because this is something that I very much stressed in my research review paper, that this is something that we need to overcome in order to treat this vast majority of neuro-associated disorders. In terms of how far my project is going now and what I'm doing with my project now, since I completed this project last summer, is that, of course, I'm doing the science symposium now, and my research review paper will soon be published in the International Youth Neuroscience Association, which is a database for high school students to publish neuroscience related research review work. And as well as I contacted some PhD students from Duke University to kind of describe that I'm very interested in CRISPR and I have written this project, and if they would be willing to help me write more work that's related to CRISPR and different neuro-associated disorders. So thank you so much. All of my sources can be found in my research paper, and that's available on my Scholar page. So of course you can go see. So thank you so much.
Thank you so much, Maria, for that awesome presentation. I had no idea that CRISPR-Cas9 could be used for ADHD treatments. That's so interesting. I have a quick question. So how widely available is CRISPR-Cas9 treatment for ADHD now, or is this really mostly theoretical?
I think that this is, from what I know, is that it's more theoretical, but due to the fact that we have the advancements in nanotechnology, we can use, for instance, the lipid-based nanoparticle capsules, where we can think of this as the CRISPR-Cas9 system, right? And usually through the blood-brain barrier, it cannot pass. And what we can do is we can encapsulate it in the nanotechnology capsule, and then it would basically imitate a molecule that would usually pass through. So I think that due to the fact that there are the nanotechnological advancements, then it will be used more efficiently, and it will be implemented more broadly. But for now, I think it's more theoretical than actually being implemented.
Awesome, thank you. We have another question in the chat, which is, great presentation. Do we currently use nanotechnology to get any drugs through the blood-brain barrier for other disorders or diseases?
From what I know, then yeah, that's actually nanotechnology is more used for to actually get drugs through. So of course, because nanotechnology, of course, it's a very broad field. So you have many different types of of the many different types of implications of nanotechnology in order to get through to the blood-brain barrier, such as like the lipid-based nano capsules and the liposomes, like different structures that you can use in order to pass through the blood-brain barrier. So yeah, I think that we can get drugs through BBB as well.
Interesting. And do you think you mentioned, really awesomely that your paper will be published in the journal, as well as you're contacting some PhD researchers at Duke. Do you plan on continuing majoring in either genetics or, psychology or neuroscience? What are your bigger research plans?
Yeah. So for sure, I want to do something more in genetics, probably also in the field of CRISPR-Cas9. I actually got really interested in CRISPR-Cas9 after doing this project last summer. And I've actually, since then, I've been doing a lot more research review articles and writing them and contacting other specialists to help me do it. And yeah, I would like to just find a correlation between genetics and neuroscience and kind of implement the different genetic therapies and genetic editing techniques into neuro-associated disorders, because I think the brain is a very developing field, and so is genetics. So I would for sure want to combine those two in the future.
Really cool. We actually have Tyler who has a question. I'm going to allow him to talk. Tyler, ask your question.
Sure.
Hi, I hope you can hear me.
Yep, we can.
Great. I was wondering if they have looked at, the use of maybe not dopamine, but something similar in animals that don't have a blood-brain barrier, like other model organisms, to prove, that something like this is possible.
No, I haven't. I haven't actually looked at that. So I for sure will look into that actually now. But yeah, I haven't looked into that into animals that don't have the BBB. Yeah, so that's for sure something interesting, and I will look into it. So thank you for the suggestion and question.
Awesome.
Great work.